Hello, everyone. Today, we are going to talk about transcriptome analysis. And we are going to study when and why do we perform this analysis. When we mean a transcriptome, we mean all the transcripts inside the cell. All the transcripts inside the cell would include mRNA, lncRNA, miRNA, circular RNA, and finally, we can talk about two other RNA which we are not really interested in. One is rRNA and the other one is tRNA. Here, if you look at all these transcripts, this transcript, the mRNA, codes for protein. Means we can say that they're coding, and all the others can be classified as non-coding. And the RNA that dominates your RNA pool is your rRNA. This dominates your RNA pool. We'll look into how we can negate this rRNA when we are estimating all the rest of the RNAs. Now, when we are looking into transcriptome analysis, the basic fundamental behind transcriptome analysis is, let's take an example. Let's say that you have a control sample and then you have an infected sample. And there is manifestation of a disease in the infected sample. And when there is manifestation, you want to find out the actual mechanism behind this manifestation. And the mechanism behind this manifestation of the disease can be best studied by transcriptome analysis. Once again, I'm repeating. See, when you have infected samples, there'll be some manifestation of a disease, and you would like to find out the mechanism behind this manifestation of the disease. And this can best be studied by transcriptome analysis, right? And for this, the primary thing that all of us should remember is we should ideally take 6 samples a group when we are actually analyzing our infected with respect to that of the control. So what are we looking at? We are basically looking at how the different molecules inside the cell would influence a particular phenotype that is observed under infection. When you say manifestation, there's a certain phenotype that is observed under infection, and you'll have to look at that. Now, for example, now let's see what people used to do earlier. People generally used to study only the expression of mRNA under a particular disease, and this was generally done by real-time PCR. And this used to be done one gene at a time. Now, the basic behind this that all of us should and must remember is a phenotype. This is the bottom line and the punchline. A phenotype is seldom determined by a single gene. What does this mean? That a phenotype is rarely determined by the expression of a single gene, Now, what does this imply? This implies that the phenotype is basically governed by the interaction of several molecules that are expressed within the cell. This study of interaction of molecules within the cell, which would actually explain the mechanism behind the phenotype, is termed as systems biology. The easiest way to study the systems biology is nothing but by transcriptome analysis. The two main tools by which you can analyze the transcriptome are: one, the microarray; two, RNA sequencing. We will talk about these two techniques in detail in the next lecture. However, before that, we will be talking about the various interactions between the molecules that we are going to actually study in the RNA sequencing analysis. So we'll have interaction of molecules, and the molecules that you generally have in hand in the transcriptome are: one, the mRNA, and this is normally analyzed in RNA sequencing. I've already explained in one of my previous lectures that through RNA sequencing, the RNA that is got is initially isolated by using oligo-DT probes, because all the mRNA, they have the poly-A tail, and you have the mRNA separated, and then go in for sequencing. Then you are going to ultimately end up, after the analysis, with differentially expressed genes. They are called as DEGs. And similarly, when you finish off your miRNA analysis, the microRNA analysis, through the pipeline, which I'm going to explain later, you will be arriving at differentially expressed microRNA. And as well, like with the LNCs, you will be finally ending up with differentially expressed LNC RNA, that is DE-LNC RNA. And similarly, for the circular RNA as well, you will be having circular, differentially expressed circular RNAs. What we are going to study today is, like, how are these molecules interacting with one another? I want everyone to be attentive over here. The mRNA once transcribed, this is the transcribed mRNA from the 5' to 3' end. This transcribed mRNA towards the 3' end has got a 3' UTR, Now, the molecule that interacts at the 3' UTR is your microRNA. The one that is given in red is microRNA, miRNA. If you have a microRNA that is binding to the 3' UTR of your transcript after expression, what happens is the ribosomes which come across the transcript for translation, when they find a heterohybrid at this particular point, these ribosomes drop off and the translation do not take place. And the translation does not take place. And if the translation does not take place, what is happening here? Though your mRNA is completely expressed over here, just because of the binding of the miRNA to the 3' UTR, there is no protein that is expressed. So miRNA expression does not correlate with your mRNA expression, but correlates with your protein expression. Because here, once again, I'm stressing this point, miRNA binds to the 3' UTR, thereby preventing that transcript to be translated into a protein. So miRNA is not influencing the expression of your mRNA. It's already expressed. And now what's happening here is just by the binding of the mRNA to the 3' UTR, your protein is not being translated. And your protein expression is ultimately affected and not your mRNA expression. So this you need to understand. So this is one kind of interaction that you'll have to remember throughout. Similarly, the next interaction that you should understand is proteins interact in the form of transcription factors binding to one another. Two, some of the proteins may help in scaffolding and chromatin reorganization, and thereby further help in several ways in the expression patterns. The most important thing is the third one is your LNC RNA, which has got umpteen number of functions. And the LNC RNA, one of the main functions is sponging of your miRNA. Sponging miRNA. What happens here? Just have a look at. If you have an LNC that sponges your microRNA, this microRNA is prevented from binding towards the 3' UTR of a gene, and the protein translation is normal. Now look at the interaction. It has just become the opposite now. What has happened? The microRNA is being sponged, and therefore the microRNA cannot bind to the 3' UTR, and the protein expression is normal. And similarly, your LNC RNA can influence gene expression in different ways. And we'll have a separate lecture on the role of LNCs later on. Now, fourth one is your circular RNA, which have got a similar role as that of your LNC RNA in sponging your microRNA. They again sponge your microRNA, thereby preventing the microRNA from binding to the 3' UTR. So here again, the point to be stressed is like the expression that you are looking at or the phenotype that you are looking at is not only because of the DEGs that are expressed over here. It is mainly because of the interaction of all the different molecules that are actually interacting with one another to result in a particular phenotype. And this study of interactions is finally termed as systems biology. Systems biology. We'll talk about RNA sequencing and microarray in the next lecture. Thank you.